Today we are going to repeat the topic of epistasis, and we are going to repeat the yesterday's example, and we are going to learn another example from your book, right? Now, if you guys remember yesterday I gave you guys the example of Bombay phenotype. Now, if I write down the definition of epistasis, it is very simple, that when one gene masks the effect of another gene present on a different chromosome or different locus, it is called as epistasis. Now, the definition feels a little bit complicated, but it is not when we understand the example. I told you guys that let's suppose that there is chromosome number 19, right? And there is chromosome number 9. The chromosome number 19, it has the gene for the antigens A or B. There is no third antigen, right? It has the gene for the antigens A and B. While chromosome number 9, it has a gene for an enzyme that produce a carbohydrate chain. That produces a carbohydrate chain. Now we know one thing, that antigens, they are glycoproteins, right? What are antigens? They are glycoproteins. And we also know a glycoprotein means that it is a conjugated molecule. You guys have learned that in detail last year in AS. And it is made by joining a carbohydrate with a protein. Now the antigens for A and B, they are made by joining this carbohydrate with this protein. Right? Now, if this gene, which is present on chromosome number 9, which is also called as gene H, if it is recessive, if this gene is what? Recessive, then what will happen? Then this carbohydrate chain will not be formed. This carbohydrate chain, it will not be formed. But this antigen, let's suppose that the person has, you see, we learned in detail yesterday about the genotype and phenotypes of the red blood cells. Now, very quickly, just let me draw. A person, if has antigen A on its red blood cells, this person will have A blood group, right? If a person has antigens B on its red blood cells, this person will have what? B blood group, right? And if a person has both A and B antigens, A and B antigens. So this person will have A, B blood group, right? So this person has A blood group. This person has B blood group. This person has AB blood group. And you guys know, you see, we learned this yesterday. If there is no antigen present on the surface of the red blood cells, then this person will have which blood group? O blood group. So this person will have O blood group. Now, this blood group or these antigen present on the surface, they are actually what? These are actually glycoproteins. These are actually what? Glycoproteins. Right? And how they are made? By joining this protein with this carbohydrate chain. Now, we have also learned, if you remember yesterday's lecture, this person can have two genotypes. This person can have two genotypes. Let me use some other color. Let me use green color to represent. This person can have the genotype IA, homozygous dominant, right? Or this person can have the genotype IA and I0. Do you remember this or not? It means that the antigen A is being produced in this person. This person who has B blood group, his genotype can be what? IB, or it can be IB, I0. Do you understand this or not? And this person who has AB blood group, his genotype will be IA, IB. And if you guys remember, this is the example of codominance. What is codominance? That both alleles are expressing themselves equally. Half of the antigens are A type and half of the antigens are B type. Both of the antigens are present at the same time in one person. So both genes are expressing themselves equally. So we call this phenomenon as codominance. Understand? While over here, the genotype is what? I0. Now, as both alleles are recessive, so no antigen is expressed, right? So this cell will not have any antigen on its surface. Now, these phenotypes will appear. These phenotypes will appear if the gene H has the genotype capital H or capital H small h. Now, if this genotype is there, then there will be production of carbohydrate chain, right? It means that this carbohydrate chain will join with the protein part of the antigen and glycoprotein will be formed. Well done. But if this gene H, it is homozygous recessive like this. If it is homozygous recessive, now what will happen? No production of enzyme. Enzyme will not be produced. When the enzyme will not be produced, then what will happen? No carbohydrate chain. This carbohydrate chain will not be there, and a complete protein part of the antigen is there, right? Protein part of the antigen is there. Now let me tell you, a person can be either IA, I0 or IA, or a person can be what? IB, I0 or can be IB. A person can be IA, IB. So what? Just a bit. These can be the genotype of a person. These can be the genotype of a person. But if he has small h, small h, small h, small h, small h, small h. Right? This is normal people. These are the people who have capital H or capital H, small h, all of them. This is another scenario where they do have dominant alleles for the antigens A and B, but the chromosome number 9, they have homozygous recessive. Now, in these people, the carbohydrate chain will not be formed. Right? Now, when the carbohydrate chain will not be formed, glycoprotein will not be formed. So, those antigens will not be expressed on the surface of the red blood cells. Now, if I draw a red blood cell for this person, for this person, for this person, even though their genotype, this person's genotype is what? A. This person's genotype is B. This person's genotype is AB. Proteins are there. But they are not displayed on the surface. Appearance will come like an O blood group. Phenotype will appear like a O blood group. But genotype is what? Either A, B or AB. Now, what has happened over here? This is the gene present on chromosome number 9. It has masked the effect of the gene which is present on chromosome number 19. So, we will call this as epistasis. Do you understand this concept now? Right? So, two genes, they are bringing a phenotype in appearance. Understand? And one gene can mask the effect of the other gene. Got it or not? Anyone has any question about this? Clear? Now, the example from your book. Now, in your book, they have given the example for the feather color. Now, feather color is also an example of what? Epistasis. They say that there is a gene called G. This gene is responsible for feather color. Right? But there is another gene, F. Now, if this gene is present in dominant condition, means that this gene can be capital F, this gene can be capital F, small f. Right? If this gene is present in these both conditions, then the color will not appear. Yes. Color can only appear if this gene is in which condition? Homozygous recessive form. Right? Now, this gene can be capital G, it can be capital G, small g, it can be small g, small g. Right? Now, there can be different combinations where the feather color can be produced. A chicken which has capital F, capital G, it will have white feathers. Now, what has happened? FG, now G produces the color. G produces the color. But this F, when it is in dominant condition, it has masked the effect of G gene. The gene that produces the color. And it has become what? White color. There is another, you see, combination can be there. Capital F, capital G, small g. Still white feathers. Even though there is what? One dominant allele is there. There is another possibility. Capital F, small f. Capital G. Now, still one dominant allele is there, right? For the F, it will still be what? White feathers. Right? There is another possibility. Capital F, capital G, small g. Still, there is one dominant allele, but there is one dominant allele for the one which is masking the effect, right? Still, it will be what? White color. But if it becomes small f, capital G, then it will be what? Colored feathers. Right? It can also be what? Small f, capital G, small g. Again, it will be what? Colored feathers. Now, interesting thing. If it is small f, small g, what it will be now? White feathers. Because obviously this is recessive, but this gene is also what? Homozygous recessive. If it is capital G or capital G, small g, then it can produce the color. So in this case, it will be what? White feathers. So in this example of the feather color for the chicken, what is it happening? That this gene F, it is masking the effect of gene G. Now both are present on different chromosomes, all at different loci. You know what is loci, right? Position of a gene on a chromosome, right? But they are affecting each other. So this is the example of what? Epistasis. Understood, everyone? Now, another thing, you see, one of the most expensive dogs in the world, Labrador, and especially the golden. Now, Labrador, they have three varieties. They have chocolate color, they have golden retriever, the one which has, like you see, this blonde color fur. That one is called as a golden retriever. That one is an expensive that is an expensive breed. That is also an example of what? Epistasis. So the fur color in the golden retriever, in the Labrador dogs, that is also maintained by two genes. Understand? And that is also an example of epistasis. Understood, everyone? Okay. Now, next thing which we are going to discuss. It does, you see, okay, I will use this much area for this example. Next one. Now, in humans, you see, till now what we were discussing, we were using simple letters. For tall plant we use just capital T, right? For short plant we use simply small t, right? But if they are discussing the genes of the humans, there is a special method to represent those genes, the human genes. Now, the gene, like one example, there is now the representation of the gene is like this. Now this is gene which is called tyrosinase gene. Now this tyrosinase gene, this is responsible for albinism. Now first of all, question is, sir, what is albinism? Now sometimes you see that black parents in African Americans or African real, right? They have a son or a daughter that is totally white, pale color, like white. Even hairs, they are blonde, very light blonde. Eyelashes, right? And eyebrows, they are also what? Blonde color, right? Very light white in color. Even the texture of the hairs can be very, very hard, like you see, or thick, like you see African people. But the skin color, every see hair color. Now, interestingly, even their retina, that is also of the very, very light color. Now, why does this happen? Now, let me tell you that in these people who are albino, now this phenomenon is called as albinism. But the person who is affected with this condition is called as an albino. In them, this gene is recessive, homozygous recessive, right? When this TYR, tyrosinase gene, it is recessive, then an enzyme is not produced that changes tyrosine, which is an amino acid, into melanin. Now, let me tell you one thing, that this tyrosine, first it is changed in a normal person I'm talking about. Right now I'm talking about normal people. Into DOPA. What is DOPA? Dihydroxyphenylalanine. This is a modified amino acid. Have you heard about alanine? It's an amino acid, right? So dihydroxyphenylalanine, a modified amino acid, right? Then this DOPA is converted into dopaquinone, another molecule, and then dopaquinone is converted into melanin. Melatonin is a hormone. Now, melatonin is a hormone that regulates sleep cycle. Now, melanin. Now, you guys may watch the advertisement of Fair and Lovely. Put the Fair and Lovely on your face, and it reduces the production of the melanin, and you become Gora, right? In 7 days, right? Now, so actually, in a normal person who can have homozygous dominant or heterozygous, right? In that person, this is the process that will be taking place. And the enzyme that converts the tyrosine to the DOPA, that is the tyrosinase enzyme. Now, if this enzyme is defective, then it means that tyrosine will not be converted into DOPA and dopaquinone. It means that melanin will not be produced. It means that the person will not have pigment in its skin. Now we have color, that is because of the pigment, that is because of the presence of melanin. That pigment, that melanin will not be present in that person's body. So the person, he will have difficulty. Now, these people, they are very easily prone to get skin cancer. Now, you see, you know that those people who are lighter of the skin, they are more exposed to the skin cancer rather than the people who have darker complexion. Another very important point about these people that I just told you few. So the iris of these people is what? Translucent, right? So as the iris is translucent, so what will happen? That these people, the light rays, they will be able to pass through the iris, right? So that will overstimulate the retina cells. And because of this, their vision is not clear. If they are in the bright light, they will be like this, always. And they have to wear the sunglasses all the time in the sunlight. At night time, they have a bit better vision, but at daytime, it is very difficult for them to see because of this. Alright? So this is one of the, you see, so these are the problems these people, they face who have this gene which is defective in them. Alright? The next thing which we are, or the next gene that we are going to discuss, that is the HBB. I don't need to explain this in detail. You already know. We have learned zillion times about this. Hemoglobin, sickle cell anemia. Hemoglobin, sickle cell anemia. Now, please pay attention. We have learned that a normal hemoglobin gene, it produces normal hemoglobin, Right? Now, who will tell me how many polypeptides are present in the hemoglobin gene? Four polypeptides. There are 2 alpha and 2 beta. If you guys see that the line for the beta I drew a bit longer. Why? Because they have more amino acids. There are 141 amino acids, 141, 146, 146. Let me mention over here. 141, 141, 146, 146. Now, in a normal beta chain, on the 6th position, there is glutamate. In the normal hemoglobin, on the 6th position, in the beta chain, there is glutamate. Glutamate is an amino acid, right? And this glutamate, it is a polar amino acid. Right? Now what happens? Point mutation. What happens? Point mutation. Just change in one nucleotide. Change in just one nucleotide causes to replace glutamate to valine. And valine is non-polar. Now, question is, when glutamate is replaced by the valine, what is the effect? Now, the effect is that this valine is non-polar and glutamate is polar. Because of that, the tertiary structure of the beta chain is disturbed. The tertiary structure of the beta chain is disturbed. So, as the tertiary structure of the beta chain will be disturbed, the proper globule will not be formed in the quaternary structure. And proper carrying capacity of the oxygen will be lost. Right? So this is because of the defective. This is because of the defective hemoglobin gene. Now remember that. We have learned that sickle cell anemia is another example of what? Codominance. That is another example of codominance. It's not the recessive gene, it is the defective gene. Remember that. And that is just because of what? Point mutation. Change in only one nucleotide at the 6th codon. At the 6th codon. You know what is a codon? Do you remember this or not? The codons that are responsible for the amino acids, right? So the 6th codon, only one nucleotide is changed.
So how is it codominance?
Yes, because both, you see, let's suppose if a person can be HbA. This person's genotype is what? Homozygous normal. A person can be HbS. Both alleles are defective. Both alleles are defective. His whole hemoglobin will be what? Defective. But a person can be what? HbA and HbS. Now, in this condition, this person will have both normal hemoglobin as well and defective hemoglobin as well. Understand? As this person has both normal and defective hemoglobin, so this is the example of codominance. Like over here, the person has what? Both antigens A and antigens B present in the same person at same time. So this is example of what? Codominance. This is also example of what? Codominance. So that's why we do not say that defective hemoglobin or sickle hemoglobin, that is a recessive allele. No. Understand? That is the example of what? Codominance. Remember this. Remember this. Understood everyone? Next example. Everyone understood this example? Yes.
What's the recessive gene?
There is no recessive. There is no recessive. Like, now, why you see over here, remember, in the blood group, for one gene there were three alleles, right? Understand, there were 3 alleles. Let me write over here, it was IA, IB and I0. Over here there are only 2 alleles, HBA and HBS. Alright? As there are only 2 alleles, there are 3 phenotypes. Here there are 3 alleles, here are 4 phenotypes. Understand? So if the number of alleles will increase for a particular gene, the number of phenotypes will also increase. Number of phenotypes will also increase. Now let me give you one example. Are there only 2 types of people in the world, black and white? No. There is a range of complexions over here, right? There is a person maybe in the class who is fairest in complexion. There is a person in the class who has darkest complexion. But there are not only 2 extremes over here, right? There is a shade of complexions over here. And that shade not only depends on the genotype, that also depends on the environmental factors. How much time you are spending in the sunlight as well. So that you have to put fair and lovely. Right? So the thing is that why we have so many range of complexions? Because your skin color is not controlled by one gene. It is not controlled by one gene. It is controlled by 3 genes. 6 alleles. Right? The person who will have all the 6 alleles recessive will be fairest. Not in his decisions, in his complexion. Right? And the person who will have all the dominant, he will be having the darkest complexion, right? So that is the thing. Now, everyone understood till here now. Yes. Now we are going to talk about another disease and another gene, Huntington gene. Yes. Now boys, this Huntington disease or Huntington gene, very interesting. This Huntington disease, it is because again of a defective gene. This is very important thing. The inheritance of this gene it is autosomal dominant inheritance. Very important point. Huntington disease, this is what? Autosomal dominant inheritance. Means that if a person gets a dominant gene, he will have the disease. If a person gets the recessive gene, he will be normal. Opposite. Whatever you have learned till now, it is opposite to that. It means that, you see, most of the diseases happen because of the presence of recessive alleles, right? For the first time, you are hearing about a disease that happens because of a dominant gene. Now, simple thing. Why do we call a gene as dominant? Because it can express itself. So if this gene will express itself, it will cause the disease. Right? Now, what happens? In this disease actually, what happens? Mental retardedness takes place. But it's not by birth. It's not by birth. It usually starts. Now, another thing. This is a progressive disorder. This is what? This is a progressive disorder. What does this mean? It means that its intensity increases with the age. Its intensity increases with the age, and usually the disease starts showing its symptoms after the age of 30. Right? Now, or after the age of 50. It's in between 30 and 50 years of age that the symptoms start appearing. The question is, sir, what are these symptoms? Now, as you see, this affects the brain. Actually, what happens? That when this gene is dominant, it produces Huntington protein. The dominant gene produces what? Huntington protein. That Huntington protein damages the parts of the brain. Now, it damages the part of the brain which is related to speech. So the person will start having slur, you see, will not be able to pronounce the words properly. The speech will become slurred. Right? Then it can also damage the motor connection with the muscles as well. So the muscle control can also be what? Lost. But remember, it is not at once. It is not at once. It is a progressive disorder. Understand? So what can be done? Now remember, as this is a genetic disorder affecting the brain, there is no cure for this.
So can't you get inhibitors?
Yes, we can. But till now they have not been able to produce that. What they can do in this, you see, that with the physiotherapy and exercise, they can help in maintaining and developing the connection. Remember that. I told you why the memory develops. I am teaching you something right now and you are learning. And it is being imprinted into your brain. You are memorizing it. What is, you see, in a computer there is a hard disk where the data is stored, right? How the data is being stored in your brain? Actually, new connections are being made, new synapses are being made in your brain. That develops the memory, right? So when physiotherapy is given, right, speech therapy is given. Now, if you have seen the one movie that got Oscar as well, that was about the King of England who was unable to speak properly. I don't remember its name now. So in that, you see, they showed that speech therapy, how it works. So if the speech therapy is given, physiotherapy is given, then the symptoms can be prolonged. We can't stop the symptoms, but we can prolong them. I was, you see, watching one YouTube video. There was an athlete who was actually what? Very well built. He was above 30 and he had Huntington's disease. Right? But he was continuously doing exercise, and he was doing exercise more than, you see, difficult exercises than a normal person. Now the question is, as I told you that the symptoms, they appear after the age of 30. Now, it's not compulsory, you see, stamped that it will be after the age of 30. For some cases, it may be developed at the age of 15 as well. Right? But most of the cases, average number of cases, they develop their symptoms after the age of 30. The question is, how do we know beforehand that this person has Huntington's disease, or this person has the defective gene before the symptoms start appearing? Can you think of any reason, any test by which we can detect the person has Huntington's disease? Anyone? No. Up till 30 years of age, no symptoms are there. But we want to find out whether the person has Huntington's disease or not.
So you check their parents if they had.
This is one of the options. We go for gene analysis. We go for genetic analysis. Understand? And another very good point by this author. Very simple thing. If one of the parents is affected there are 50% chances that their offspring will be also affected. Why? Because this is a dominant trait. Now remember, yesterday we talked about, day before yesterday we talked about hemophilia, right? It was a recessive inheritance, right? A person became hemophilic only when a recessive allele comes, right? In homozygous condition or hemizygous condition. Remember that or not? Over here, let me show you. A person who is capital H, small h, this person will have Huntington disease. A person who is capital H will have Huntington disease. A person can only be normal if the person is small h. Understand? So if a person, just a minute, if a person's parents, now you see, usually the marriages, they can take place before the age of 30, right? Let's suppose that a man who has Huntington's genes, right? And he is homozygous dominant for that. He marries a woman who is 100% normal, like this. All of their offsprings will have Huntington's disease. Right? Why? Because the father is giving half, because this is autosomal. Remember one more thing, this is autosomal. Half of the autosomes are coming from the father, half are coming from the mother. So 100% chances that his offsprings, they will be what? Huntington's disease. But let's suppose if the father is what? He is heterozygous. Then the chances become what? 50%. If this allele goes into the offspring will be normal. Because the man married a normal woman. Right? But if this allele, the dominant one, goes into the offsprings, then the offspring will be what? Huntington's disease. Right? Now, this is one example. Let me give you another example which is not in your syllabus. The name of that disease that we mentioned over here, Duchenne muscular dystrophy. Duchenne muscular dystrophy. Now, what is the meaning of dystrophy? Destruction. Yes. Now, again, this is also a dominant autosomal disease. This inheritance of this disease is also dominant autosomal. A person having the dominant gene, his muscles, they will progressively be destroyed. Now, these people, they live maximum 18 to 19 years of the age. Now what happens when they are born? After that, right after that, you see, when this disease strikes, the muscles degeneration starts, and a stage reaches when the diaphragm stops working. When the diaphragm will stop working, eventually what will happen? Breathing will not take place, and that can take place. These people, they become bind to the wheelchairs. They can't do their daily activities. They have to be dependent on the other people. Understand? So this is another example of what? Autosomal dominant inheritance. Right? So if a dominant, just like this, if a person will be, let's say capital D, that person will have disease. Capital D, small d will have disease. Small d will be normal. Now, these people, they are usually unable to pass on the genes. Why? They die early age. But in some communities, that the marriages do take place at a very early age, right? So that is the reason that this gene is still existing in the populations of the humans. Now, inshallah, when we learn the next chapter, we'll talk about one thing. What is the thing that we'll talk about? Yes? No, very rare. These are rare. No, but you see, but when the people they will get the disease, they will die, so they will not pass on. Only very few chances of passing on. And another very important point, very good question. Another very important point. These diseases, they may be passed on from the parents to offsprings, but these diseases are still existing because the mutation is still taking place. Right? So mutation can also cause these diseases. Mutation can also cause these diseases. Understood, everyone? Now, so I was going to say something
About next chapter.
about
The next
yes, about the next chapter. We'll talk about if there is a trait that is bad for a species, that trait is wiped out of the population. It's removed. But yes, natural selection, very good, right? But still some of those traits still exist because of either mutation or, like in this case, providing a benefit. Right? Because those people who are HbA, HbS, they are resistant to malaria. They are resistant to malaria. Now, what is next thing, please? The next thing which we are going to talk about, that is factor 8 gene. We have discussed this, right? Factor 8 gene. I told you guys that the factor 8 is a protein that helps in clotting. Remember this or not? Rather, I told you that there are 3 factors, right? Factor 8, factor 9 and factor 11. I told you guys that the factor 8 and factor 9, its genes are present on X chromosome. Well, factor 11, its genes are present on autosomes. Now please pay attention. This factor 8 gene, this protein, is actually produced in hepatocytes. The last topic of this chapter, which we will discuss, that is the lac operon. That will be about discussion. The genes, they get turned on when they are required. When they are required. When they are not required, they are turned off. Like right now we are studying in the biology lab, right? So we have turned on the lights, we have turned on the AC. When we will leave, no one will be here. Is there any use of turning on the AC and the lights? No. So we turn off, right? But rest of the school, where the classes are in use, the lights and the ACs, they will be on, right? Same, the genes, all of them, they are not turned on. We have learned that in detail last year. Totipotency, pluripotency and multipotency. Remember that or not? Totipotency is that when all the genes in a cell are on, active. Like a stem cell or like a zygote. Remember this or not? So these hepatocytes, in them, this factor 8 protein is being produced. This gene is on over there, it's active over there, right? Now, if this protein is not being produced, if this gene is recessive, now remember, I have taught you this in detail. A person who has XX, a female, capital H, she will be able to produce this protein. A person female who is X capital H, small h, again, she has the genes to produce, the dominant allele to produce the protein. A female who is small h, X small h, she will be hemophilic. She will be hemophilic. This factor 8 protein will not be produced. Because of that, the blood clotting will not take place. So person can bleed to death. Even small cuts. Even small cuts. While in men, we have discussed it can be X capital H Y or X small h y. A person who is X capital H Y will be normal. A person who is X small h y will be hemophilic. Now, again, this is a genetic disorder. Is there any treatment for this? Yes. Why there is treatment for this? Because if we inject a person with this factor 8 protein, then if he gets a cut, he can clot his blood. He can clot his blood. So, and remember one more thing, as by the process of genetic engineering and biotechnology, we can produce proteins, right? The process is expensive, these proteins are expensive, right? But still they are available, right? And they are given to the patients and they are spending their normal lives. Just 1 minute. Now, if you guys remember, I told you older Prince Charles, nowadays King Charles, he has hemophilia. He is spending a normal life, right? Why is he able to spend the normal life? Because of the breakthrough in the genetic technologies, right? That this is available. This protein is available. And one more thing, just a minute, that you see, before they were having cousin marriages. Now all their kids, they are marrying out of their families, right? To decrease, to dilute this effect of all the genetic disabilities that have been occurring in their family, right? Yes. No, because this is just a protein. It's not an antigen, right? And in a normal person, these are present. In a normal person, these are present, right? So it will not be. Understood? Everyone understood this. And second thing, that it is being injected into the blood, right? And these actually produced were in the hepatocytes over there. Alright? Everyone understood this? So I'm stopping the lecture over here, right? We have only about 7 minutes left, right?